genetic drift and mating systems and evolution and inbreeding. Haldane, who was studying genetic linkage, natural selection and evolution. And Ron Fisher, who was actually a statistician and a geneticist, and who is the person that helped reconcile how genetics, which at the time was known to be deterministic in terms of you have this trait or you don't, how that can be applied to factors that are to continuous traits like height or blood pressure. So he explained how by having multiple genetic factors, you can end up with a continuous normal distribution of values for a trait. I'm going to start with some very basic things, but I just want to make sure we're all on the same page when we're talking about every concept. You know that the DNA in humans is broken up on chromosomes, and each chromosome really holds one long strand, one continuous long strand of DNA. And we are diploid, which means that for each chromosome we have two versions of it that contain the same genes and are very similar to each other, but one of them comes from our mother and the other one comes from our father, and there are certain locations where they might have differences between them in the sequence. Each human cell has two, because we have two genomes, the maternal and paternal, times 3 billion, more or less, nucleotides. And just some interesting facts. If you take the strand of DNA, it's 2 meters in length from one single cell. So if you calculate for about 30 trillion cells in the body, the length of the DNA in our body can take us to the sun and back 200 times. So that's a very long stretch of DNA. Has anybody extracted DNA in the lab? A number of people, I see. So you know that when you do the last step, you see, actually, when you have DNA which is very dense, you put the pipette in and you pull it out and you see long, like strand. That's the DNA holding the water with it as it comes out of the solution. And also when you add ethanol to precipitate the DNA, you see like a hairball coming out. That's the standard DNA coming out. So the human genome, as I said, we have two genomes, one maternal, one paternal, and they're not identical. And when the paternal and maternal chromosomes are different, we say that this site is heterozygous. And the sequence of each one of the two genomes is called the allele. So the maternal allele and the paternal allele. Caucasian individuals are heterozygous at about 3 million sites, and that's more than twofold higher for African individuals, and this is because Africans are a much older population. They've been on Earth much longer before the Caucasians broke off and went north. So they have accumulated a lot more variation. So in Caucasians, there's about one difference every 1,000 base pairs, and twice as much in Africans. And Asians are about the same with Caucasians. There's, of all those variants, about 10 million are common. They have a frequency of 1% or more. And when a variant has a frequency of 1% or more, we call it the singular, we call it polymorphism. So we call it the single nucleotide polymorphism, which is abbreviated in SNPs. If it's less than 1%, we prefer to call it a variant, a single nucleotide variant, or an SNP. And as I said, each possible sequence are a variant, and the variant site is an allele. Now, as I said, each chromosome is one long strand of DNA, and the combination of uninterrupted sequence on one strand of DNA is called the haplotype. So each chromosome is practically the haplotype. So we each have one, for each chromosome we each have one maternal and one paternal haplotype. And during meiosis, these haplotypes are broken and rearranged by crossing over. So meiosis happens in our parent. So although my mother could have these two haplotypes here, we might inherit one of these chromatids which has a mix of maternal and paternal haplotypes. So this way it gets mixed in every generation. I'm assuming you know all this stuff from your biology classes, but I think it's important this is very basic and important concept to know. So yeah, for each chromosome, we have a mix of the two parents, but not necessarily the two grandparents. Now, I'm sure you have heard the concept of haplotype block. So I said the haplotype is the entire chromosome, but you probably know that haplotype blocks are like 20 kb long. They're not much longer than that. So the haplotype blocks are pieces of the haplotype that tend to be preserved and unbroken by recombination. And we observe that by seeing that the same combination of alleles is found in many individuals in the population, more so than would be expected if they were independently inherited. So it reflects historical rates of crossing over, and it's a population genetics concept, and it signifies that the combinations of alleles within different variants are observed more frequently together. They're correlated with each other in what we call linked disequilibrium, which we will talk about in more detail. And here it is. So the correlation between nearby variants is called linked disequilibrium, which means that the occurrence of specific alleles in the same haplotype is more frequent than is expected by chance. And how does the linked disequilibrium get created? Why are they found together more than expected? So every new variant has its history somewhere in the past by some mutation that happened. And that mutation happens on a specific strand of DNA. And we said a strand of DNA is a haplotype, all the different alleles it might have, that's a haplotype. So when that new variant happens, so here with the mutation in this G nucleotide, creating a T, that T is not going to be on both haplotypes that are in the population or on the not large number of haplotypes that might be in the population. It's going to be on the haplotype where the mutation happens, which is this haplotype in this case. So that G is going to change to T, and that T is only going to be present together with the CPAAT on that haplotype. The other combinations, so the T is never going to be seen with the G next to it. Okay? And this will continue until it's broken by recombination. So, as I said, so our new allele here, I believe, was the T here, and it was always found with the T next to it. But if during a meiosis of one of the people that have offspring, it crosses with the other, the maternal and paternal haplotype cross, the grandmaternal and grandpaternal haplotype cross in the mother of the upcoming individual, then you have that T suddenly also found in combination with the A. So now the linked disequilibrium is starting to decay. So while before, in the previous situation, we could be sure that when you have the T in that variant, the variant next door will also have a T. After the recombination, that's no longer a certainty. It can also occur with the A. Okay? So that's how LB is created. It's created by the new mutations, and then it's also created in other ways that we'll mention going down the road, like group bottlenecks where only specific haplotypes make it through, or selection which selects for specific haplotypes. But the origins of the linkage evolution from the new mutations that get into this image. Now, in order to understand how LB is studied and measured, we have to talk about the three major probability rules. I don't know if you've taken any probability, but these are very, very basic things that I think that you should all know. So the three rules are the addition rule that says that the probability of two events, the probability that any of two events happens, either event A or event B, equals the probabilities of each of those two events minus the probability of both events. The reason we exclude the probability of both events is that if you have both, it's not that you can have both in two different ways. So we don't want to double count the A and B that happens independently. In other words, if you have a 50% chance of getting COVID and a 50% chance of having hypertension, I'm just pulling things out of that, then the probability of getting one or the other, it's not going to be 50% plus 50%, 100%. It's going to be a 50% chance that you'll have one. It's going to be a 50% chance that you have one, a 50% chance that you have the other, and then minus a 25% chance where you're going to have both, because there's also a 25% chance that you get none. And this comes from the multiplication rule, which says that the probability of having two events both happen is the product of their probabilities. So again, you have 50% chance of COVID, 50% chance of hypertension. Having the both is 25%. Having one or the other is the other 50%. 25% is not. Okay? So this is the rule we're going to use the most. The other rule is mostly about comprehension of the concepts, and it says that the probability of something not happening is 1 minus the probability of that happening. So it says that something will either happen or will not happen. There's no intermediates of that sort of happen. So this brings us to how we measure LD, how we measure the correlation between two variants and see how strong linkage this equilibrium is. And here I'm introducing 2x2 tables. I think you must have seen 2x2 tables before. So here we have two SNPs, SNP1 and SNP2, and they have allele frequencies P and Q, P1 and Q1 for SNP1, P2 and Q2 for SNP2. If you genotype both, you expect to find them having the A allele, let's say, for both of them. P1 times P2, that's the product of the two probabilities. The probability of having the A allele of SNP1 is P1, of SNP2 is P2, having both is P1 times P2. Okay? The same way, the probability of having the two alternative alleles, it's Q1 times Q2. And the probability of having the reference and the alternative is Q1, this one is the alternative, times P2, which is the reference of the other SNP, right? And then there's the probability of having the other combination, which is the P2, Q2. So that's what you expect to find when you study two different variants and you study the genotypes, the combination of genotypes. However, when you actually measure them and see how often you find every combination, you might see that the combination of the two reference alleles might actually be increased by some amount that we say we call D. And D is part of how we measure linkage equilibrium. It means that's how much more than expected the frequencies of the combination of these two alleles. Now, because the allele of SNP2, because the allele of the reference allele, the reference of the SNP2 has to stay and remain P2, then we know that if this is increased by D, this will have to be decreased by D in order for them to still adapt to P2. So if you add P1, P2, and Q1 and P2, because P1 and Q1 add up to 1, this will add up to P2. So whatever you add here has to be subtracted from there. And similarly has to be subtracted from there. And because of that, it will also be adding on the diagonal. Does that make sense? Okay. So this is D, and D tells us how deviant from the expectation, or deviant for difference, whatever you want to call it, how different from the expectation is the observation of how alleles happen. Now, so D is the difference between the combination of alleles that we observe and the combination. I'm not going to get into that because I don't think that's that. But what I want to say is that D has a maximum value it can take. And why does it have a maximum value? Because none of these cells can be less than 0. You cannot measure negative number of alleles, right? So because you cannot measure negative number of alleles, D cannot be larger than these two frequencies. The SNP1 alternative value frequency times the SNP2 reference frequency. And because it's also present in this cell, it cannot be larger than this or larger than this. So that means there's a maximum value that D can take for any given frequency of alleles for two SNPs that we're looking for frequency. So the D maximum is the minimum of these two frequencies. That's what this equation is saying. Now, this is where we introduce the D prime, and D prime is something you probably have heard if you read any papers that talk about Lagrange equilibrium. So D prime is simply what fraction of D max is D. So if say, okay, D can be as much as 0.3. So this is so D max is 0.3. And we observe a D that is 2. So what is D prime? D prime is 2 over 3, so 0.6666. That's D prime. Now, one thing that's key here is that when D prime is 1, what this means is that D is D maximum. So this means that the D we observe is as high as it can get for the given frequency of allele. What this means when you plug it back into the table is that because D is the maximum value, it means that either this or that cell, one of those cells is 0, because that's where it's actually equal to its maximum value. And what that further means, if one of those cells is 0, this means that these specific alleles, so if this is 0, this means that the reference allele of SNP2 and the alternative allele of SNP1 never occur together in the same haplotype. So what that means, going back to what I was showing earlier, is that there's only 3 haplotypes in the population. The fourth does not exist, we never see it. So what that further means is that presumably there has never been recombination historically between those two SNPs, because LD was never broken. Or if recombination happened, it didn't happen between those two alleles. Does that make sense? And it makes sense to you guys.
Or it was selected.
What's that?
Again. Was it selected again?
Or it was selected again. There's other ways to get there. And so this, when this happens, when there's only 3 haplotypes, we call this complete frequency equilibrium. This yes.
If we have negative D value, how can we define D max?
If we have negative D.
Negative D.
So here I chose to put the positive D here, but this could be negative.
Okay.
But if this is negative, this
Then
will
the
be
D
positive.
max will be.
The next will be positive,
Yeah.
this will be positive, and that will be negative. So the whole thing will switch. Okay, so some of the in half the table will be positive, in half the table, the diagonals will be negative. Okay? So when there's only 3 haplotypes, we call this complete LD. So when B prime is 1, we call this complete LD, unbroken LD. But there's also what you might refer to another measure of LD is the R square, which the R square is simply the correlation between alleles, the statistical correlation between alleles. And that can also take a value between 0 and 1, and the maximum value will be 1, which means that there's perfect correlation. This means that if I know the genotype of one SNP, I can predict the genotype of the other SNP. So if that is the case, this would mean that there's no longer 3 haplotypes, but there's only 2 haplotypes, because if the 2 alleles, one allele finds itself with the other, and the other finds itself with the alternative. So there's only 2 haplotypes. This means that the frequency of the 2 SNPs has to be identical as well. But how can this come along? And one way this would come along is by changing the frequency of haplotypes. So because of various reasons, the haplotypes can change. It could be selection, it could be drift, it could be any of the reasons that frequency changes, as we will discuss further. So let's say that we started with the 20, 79, and 1% haplotypes, and I'm telling this to be very small because this is from the previous slide, the one that happened with the new mutation. So it started small, but for some reason it decreased frequency at the expense of the other haplotype, which went all the way to 0. So since that went all the way to 0, we're now left with 2 haplotypes, frequencies 30% and 70%, and perfect linkage equilibrium. If one SNP is T, we know the other is T. If one SNP is G, we know the other is A. Okay? So this is perfect linkage equilibrium. And in that case, we don't need to genotype both SNPs, because once we know the genotypes of one, we know the genotypes of the other as well. And this happens quite frequently in SNPs that are very close to each other. Now, all this, the fact that linkage equilibrium is affected by it's affected by mutations, by new mutations that happen at the age of the mutation. How long has this new mutation been in the genome? It's affected by recombination rate, but it's also affected by allele frequency changes and selection and all that. So the overall picture of LD in the genome becomes pretty complicated, and it's not really that straightforward. We can measure LD reliably between 2 SNPs, but when we come to a stretch of SNPs, you see here, this is LD. So every little one of those lines that you cannot read is the name of the SNP. So you need to know that each one is a SNP. The physical location is up above, just for reference. And below that, you get with color coding, and this is for D prime. I'm sorry, this is for D prime. You get how strong the correlation of the 2 SNPs, how strong the LD between the 2 SNPs is. So if you go between the second and third SNP, this is red, it means a very strong correlation. If you see something more pink like that, it's still strong, but it's weaker than the red. If you see purple, this purple color, whatever that is, it means that there's not enough information to know how much correlation there is. This seems to be correlation, but statistically not significant. It usually reflects low frequency of alleles of one of the 2 SNPs. And if it's white, this means there is no LD. So if you look back and take the big picture, you will see that there seems that there is a triangle here. So these SNPs seem to be in LD with each other. This seems to be a recombination hotspot where the LD is broken and it doesn't continue. And then there's another block here. But this is oversimplified because although there's this big block here, if you look at this white area here, which means that there's SNPs here and there within that block that are not correlated. And this reflects the various dynamics of the genome as it changes frequencies and as some variants. So it's possible that all the variants that are correlated with each other but not with those are younger, so they are still together, where the others are older and they reflect older recombination. So just to summarize, we said it's created by the generation of region-specific haplotypes, which can then further change frequencies for a variety of different reasons that we'll talk about when we talk about allele frequencies. It's broken by a combination which shaped with the selection, results in correlations, generates haplotype block, which is a statistical concept. The haplotype is just all the variants in the same strand. The block is a variance where is a part of that strand where they tend to be inherited together. And yes, this is just stressing in that if they're on the same strand, they are on the same haplotype. They might not be on the same haplotype block. Just I want you to have those two concepts separate, because later when we look at linkage, we'll be talking about haplotypes that are not haplotype blocks, they're just haplotypes. The consequences is that when we do association studies, for example, we can genotype a lot less SNPs. People routinely genotype 2 million SNPs, although I said there's 10 million SNPs in the genome. Then common variants, people genotype just a couple of million, because Linkers equilibrium allows them to calculate the genotypes of all the other SNPs through a process called imputation, and they don't need to genotype every single one. So that saves a lot of the cost of effort. Now, there is a downside to this, and I have here what's called a Manhattan plot, which is the results of the genome-wide association study, where every SNP is on the axis across all the chromosomes, and this is the negative log 10 of the p-value, so whatever makes above that line is considered significant. So these are all, there's a bunch of like 5 different signals here that seem to be real association, but there's not, so this association, there's however many 10 SNPs there that are all associated with disease, and we can't know which one is driving. We need to do further work in order to figure out which is driving. We'll talk about that next week. Now, I talked about SNPs so far, and SNPs are very popular. The main reason is because we can genotype them really well and easy, although they're not always the best for things that we want to do. As I said, they have an allele frequency of more than one called SNPs, because the polymorphism has to have, the polymorphism is called a variation of this frequency more than 1%. The others are polymorphic, can be polymorphic as well if they're frequent. But because the term SNPs is used commonly, I want to differentiate. There could be insertions, deletions of small sequences that for an entire set of nucleotides can be missing from one haplotype and be present in another, can be missing for some people and be present in other people. There's micro and microsatellites, which is short repeats, a couple base pairs, 5 base pairs, 10 base pairs that repeat themselves many times in the genome, and they can have a variation in how many times they repeat themselves. Especially microsatellites used to be very popular in order of the Linkers studies, because they have a lot of advantages in terms of heterogeneity, and we'll talk about that next week as well. They can be copy number variants, which is a similar concept, except here we'll talk about big stretches of DNA that can be present 2 or 3 or 10 times in different people. They can be inverted, so the whole piece of DNA is there, but it's inverted. There is a very common inversion on chromosome 17 that's about present in 20% of European people. I don't think it's, well, if it's present in other populations, it's because it's because of migration or the genetic flow and all that, but not, but it originated in Europe clearly, and it creates a specific pattern of LB around that area, which also is where the tau gene is located, the gene important for Parkinson's disease. And there's also transposable elements that could be present or absent in different parts of the genome. And Heik's agent, who was with us until recently, passed away, but he was studying transposable elements, started here at Hopkins, left for a while and came back. And this still would be important for disease, but they're harder to find in studies, so one needs to target finding them in order to study. Now, when we study population genetics, we use what is called an idealized population. So we make certain assumptions about the population. And these are actually, all of them are wrong. They don't really hold in real population. But the reason we want them is because they allow us to determine what the expectation is in order what we should observe if these rules were there. And then if we don't observe that, then we can make conclusions about why we don't observe that, what rules are broken in this situation. So these assumptions are, well, we say diploid because we study diploid population. Other population geneticists might use haploid or tetraploid or other ploids. We assume only sexual reproduction, again, because we study humans. We assume that the generations are non-overlapping, so with one generation and the next is not intermarriages and all that. We assume that mating is at random. There's no preference there's no preference that has any basis in genetics for choosing your mate, which I'm pretty sure is also not true. That we assume that allele frequencies are equal between males and females. We assume that there's no migration, gene flow, or admixture, so that people stay put where they are and they don't move around and spread the DNA in other places. We assume that there's no new mutation, which we also know is not true. And we assume that there's no selection. So if we assume all these things, then we can come up with certain rules. And one rule you know very well, I believe, is Hardy-Weinberg equilibrium, which calculates what the expected genotype frequencies are when we know the allele frequencies. So we can take the population, genotype them all, find what the allele frequencies are, and then calculate based on the Hardy-Weinberg equation which says that if P is the reference allele, Q is the alternative allele, then the homozygous for P should have a frequency of P squared. The homozygous for the reference is the frequency of P squared. The homozygous for the alternative should have a frequency of Q squared, and the heterozygous should have the frequency of 2 times P times Q. Now, where this comes from is again this 2x2 table, which is a little different than the previous 2x2 table in terms of what it means here, where you have the female gametes and the male gametes, and you try to figure out how often are they expected to come together. Again, if mating is random and frequency in males and females are the same and all that. So, if you have alleles A, big A, little a for the male and the female with frequencies of P and Q. See here there's no P1 and P1 is the same signature, the same frequency, the P is the same for both. Then you expect the A gamete to find the big A gamete to find the big A gamete of the male in P square frequency that many times. And the alternative is Q squared, and the alternative versus reference is P times Q, reference versus alternative is P times Q, so this twice P times Q, and this gives you the Hardy-Weinberg equilibrium equation. That's what you expect. But now imagine that you have, for an example of how Hardy-Weinberg can be broken, imagine that you have two populations that you don't know are two populations. One of them has 100% frequency of allele A, they're all homozygous. The other one has 100% frequency of allele little a, they're 100% homozygous for the alternative allele. You put them all together, you genotype them, and you get, although you get a frequency of 50% of one, 50% of the other, because if you have equal numbers of the two populations, so the allele frequency will be 50%, 50%. So you, based on that, you would expect 25%, 50%, and 25% genotypes, But the genotypes, in fact, you see that are 100% homozygous for one, like half of them are 100% for it, and the other one, so 50% homozygous for one, 50% homozygous for the other. So Hardy-Weinberg is violated, and you know immediately that something is going on. So Hardy-Weinberg is one way that you can immediately see admixture, but you can also see in other ways. I think I have specific slides for that. Well, I'll say it now and then. You can also use it to quality control your data, because if you have a platform with genotyping SNPs, and for some reason your platform calls too many heterozygotes for a SNP because it's got a problem, then it will be out of Hardy-Weinberg, and you will know that this is a bad SNP. The difference would be that if it's because of admixture, it's not going to be just one bad SNP, it's going to be a lot of SNPs that show that they're out of Hardy-Weinberg. While if it's a bad marker on the array, then it's going to be that one and then another one somewhere else. So if the population has an infinite size, as population genetics assumes, the study that we're doing assumes, then it can be shown, and I'm not going to go into detail, but it can be shown that the frequencies of alleles and the frequencies of genotypes will be preserved if it's an infinite population. Random mating and all that, the frequency will be preserved forever. They will never change. However, in a limited population, for let's say 100 people, even if the p-square is 0.635, you realize that you cannot have 63.5 people with a specific genotype. You're going to have either 62 and 64, you're going to have something other than 62.5. In fact, it can vary quite a bit. So in every generation it will change. And this is, because this change, it's the small population child size makes it necessarily change, and the smaller it is, the bigger the change can be. And that leads to genetic drift, which I believe we're going to talk immediately after this. So this I talked about, and I'm going to talk about again in the section about association. So I'm going to skip it right now, but it's how we can use language in the data cleaning. Now in real population, as I said, there's non-random mating, and there's often admixture resulting from migration and the non-random mating. If a new population comes and joins another population, initially there's admixture. But if there is random mating between the two populations, then soon the allele frequencies get equalized, they get balanced out, and you have again what can we consider one non-admixed population. However, in real world, we know that this is usually not happening. We know that migrants go to the country, and they usually go and find their own people, and they will get married with their own people, and they will have children, and they will stay like that for a number of generations before they start really freely mixing with others. So, as I said, Hardy-Weinberg is strongly violated in case of admixture, and that is a problem in TOS that we will address next week. So now we're going to start talking about allele frequencies and how they change, because that's an important concept in population genetics. So when there is limited size of the population, there's genetic drift. We're going to talk about next how it happens, why it happens. But that leads to big allele changes or frequency changes over time. When there's selection, either because disease takes out a lot of people choosing specific genotypes, or even randomly eliminating specific genotypes, that's another thing that will change allele frequencies. But also if an allele is associated with increased fecundity, or having more children, then that allele will increase the population, because the people that have it are going to have more children. There's bottlenecks where the population goes through some kind of selection. It could be a volcano that exploded and killed half the island population, or any of those things. or founder population effects, where new population goes and colonizes the place. But this population is only a small fraction of the original population, so their frequencies are those of the select people at best. And then there's migration and regime flow. Now, genetic drift, why do frequencies change? Let's assume a very limited population of 8 people, and the variant that only one of these people has. And these people have offspring, these couples, I have those couples here, they have offspring, and it so happens that this one person had a couple of offspring with that allele. So that increased the frequency directly from it because it's such a small population. The frequency from the 12% that it was here became 30% because of the 3 offspring this person had. And if that continues one more generation, it can become 46% in the next generation. And this is because just because of chance, these persons happen to pass that allele very often. If the population was not that small, that would be absorbed, it would not happen so fast. And here on the right side, I have a simulation. This is a script that is publicly available. You can run a simulation just to see if I can look it up and try to find it if anybody's interested. So, but here I asked it to give me 50 alleles, and starting at the 5% allele frequency, and give me 500 generations. This is T for time, so there's a number of generations. So in every generation, it takes this 50 markers, each one represented by a line, and it sees what the frequency can be. That's by chance, without any selection. Just count what the frequency will be in the next generation, and then the next generation. That's 300 people that should like the So that's 300 people already. That's the people we need. And you see that we started with 50. Many of them get eliminated really early on. When we start at 5%, it just happened not to be passed on to the bone. Others, however, start going up and reach frequencies of even above 50%. And actually, some of them, if I continue with more generations, they could go up and reach 100%. And this is purely by chance, purely by what gets moved down to the next generation by chance. No selection, no other variable. And here I'm doing the same thing with different numbers of people. This is the 300, this is the same, it's not the same plot, but it's the same parameters that I showed before. This is with 30, and you see with 30, it really goes fast. But then with 3,000, it goes much slower over the 200 generation. And with 30,000, it stays stable much more. Yet it still drifts, it still moves. So imagine this is 200 generations, imagine even more generations, this change can be significant. So this is drift. I think this might be some good titles. Here I'm saying again. Also, this is a new mutation created, as I showed earlier, that creates one haplotype. It's subject to drift, bottlenecks, or selection. And then I'm going to talk about bottlenecks. Well, I'm not going to talk much, I'm just going to show this slide, which says that you might have a population that has a number of different haplotypes or genotypes, shown here by different colors. But something happens and the population is reduced to much fewer people, and that's the bottleneck. And of these fewer people, only 2, which might have been the most common in the original population, but only 2 of these haplotypes get preserved, and then the population expands, might reach the original size, but many alleles are lost because of that bottleneck. Now, selection is what happens when the probability of an allele to be transmitted to the next generation is not 50%, as one would expect by chance, but it's different than 50%. And it can be a number, there can be a number of difference for that. There can be that anybody that carries that allele might have a higher frequency of pre-reproductive death or disability and never get to reproduce. It might have, these people have reduced, the people carry this allele have reduced fertility, in which case this allele doesn't get passed on. And reduced doesn't have to be by much. It could be like a 10% reduction in fertility. That's enough to change the dynamics of the percentage of allele, and within a few generations change the frequency. It could be that the variant is detrimental to the sperm or egg, so there could be, the person might be fine, fertilization might happen just fine, but maybe many of these fetuses occur because they might be aborted, so they are able to make it the next generation. And it leads to a systematic change in the slope of the drift curve, disappearance of unfavored alleles, dominance of the favored alleles. And I believe I have this graph here again. There's no difference from what you saw before. I just want you to imagine what would happen if the entire slope, if this is the average slope of all the variants, if you move it like this, then these would not go as high, more of them would disappear. It would change, so that selection would change the frequencies at the end. However, drift can actually make even an unfavorable mutation, and therefore an unfavorable variant persist, as long as the selection is not very strong. And we see that with some complex disorders, including schizophrenia, which I'll talk a little bit about. That although you would expect them to be unfavorable, because the effect of an allele on the risk is very small, drift can still keep it afloat. It can keep going for a long time. So the types of selection, you can see them in two ways. One is by the effect on the trait, in which case you can say that if one extreme is favored, then you call directional selection towards the direction of the extreme that is favored. If the intermediate phenotypes are favored over both extremes, that would be a stabilizing or balancing selection, where you actually going to go towards a 50% frequency so that the heterozygotes stay at their maximum level. If both extremes are favored over the intermediate extreme, that might actually lead to speciation, might lead to the creation of different species because of selection towards different directions or different subpopulations. It can also be seen as by its effect on allelic diversity, as purifying selection, either negative or positive, where a specific allele is selected for or against. Or balancing, which, so this is equivalent to stabilizing, where the heterozygote has an advantage, so there's a negative selection depending on the frequency. And there's an example of that here. I don't know if you've heard this example before, it's kind of classic in genetics, but on the map on the left, you see the distribution of malaria across the world. You see it's mostly around the equator in the southern hemisphere. It has a lot to do with climate. It has a lot to do with where the mosquito that passes malaria lives. On the right side, you see the frequency of sickle cell disease across the world. And you see that it's actually very similar to the distribution of malaria. And this could, of course, be by chance, but we know that heterozygotes for sickle cell disease, although they don't get for the sickle cell disease trait, although they don't get sickle cell disease, so they're not sick, but they have high resistance to malaria. They don't get as sick from malaria as people that don't have that allele. So in the countries where malaria exists, heterozygotes for sickle cell disease have an advantage over both homozygotes. So you have an increase in the frequency of the sickle cell disease. Selection sometimes leads to selective sweeps where an allele is completely lost to the population or another allele rises to 100% frequency because it's so much favored. And that can be studied in many ways. You see that you can look at allele frequencies and how they change. It will increase allele frequencies of the alleles that are linked to that to the favored allele. So not only the allele that itself, but also whatever is be with it will increase in frequency because the whole haplotype will be selected for. It reduces diversity because variation is locked. I talked about locally, so there's less diversity of genotypes locally because the selection of the specific haplotype will be at the expense of all others. And it will lead to excess of low frequency alleles. So there will be those selected alleles that are all going to be high frequency and all the others are going to be low frequency. So the frequency distribution is not what would be expected if there was no selection going on. So I briefly talked about speciation. So speciation is what happens when you have, so the definition of two species is that they cannot reproduce with each other. If they can reproduce with each other and give fertile offspring, then they are the same species. And I say that because there's also the hybrids is like donkeys and horses can reproduce and they give mules, but mules cannot reproduce. So donkeys and horses are close, but they're not the same species because they cannot give offspring that can reproduce. So if two populations of the same species experience reproductive isolation, so if they split, go to different places, stay there for a long time, and there's no, or if there is a disruption of gene flow for a subpopulation, so if one population goes somewhere and there's no longer gene flow from the original population, then they will acquire new mutations. There will be genetic drift. The frequency of many things will change, and eventually they will also be subject to different selective pressures. So eventually they might become so different from the population that they started with that they now can no longer mate with these people, and that will mostly happen when you have chromosomal rearrangements and things like that that will prevent fertilization. So they can actually become a different species and can only reproduce among themselves. Okay, so I'm going to show you an example for schizophrenia because I think it's kind of striking how we can see this happening in real life, how selection can shape variation. So patients with schizophrenia have half the number of offspring than the healthy population. And this is a huge negative selection, so having half the number of offspring. So you would expect that whatever alleles cause schizophrenia should be under a very strong negative selection. So what is it, why is it the schizophrenia still persists for in the population over the century? So there's a few possible answers, and I'm going to show how all three of them are actually likely to be happening. So it could be that there's a lot of replacement, replenishment by new mutations. It could be that there's very small effect on the risk, in which case genetic drift can keep them afloat, and in combination, of course, with the new mutations, they will stay in the population for a long time. Or there could be balancing selection, or there could be an advantage to the heterozygous for those alleles that we don't know about. And of course, as I said, it could be all of the above, like we eat all of the above. So replenishment, for one thing, we know that there's 250 was the last tally of genome-wide significant genes that are involved in increasing the risk of schizophrenia. So this, if there's a similar number of genes that are involved, this means that there's a huge possibility of mutations, of new mutations in these genes. So they could completely be replenished. What's more, we know in schizophrenia that there's a lot of the associated variants are actually big copy number variations. And these are often produced because there is tandem sequences on each side of the copy number variation that leads to an equal recombination. So there's a lot of de novo events. a lot of de novo deletions or insertions that lead to schizophrenia. And that we actually know, and there's a table with a list of all those that we know about, and their statistical support is pretty high. Another thing I said was very small effect on the risk. We know that is true. On the left, again, that's from the GWAS, but the risk of all those hits from the GWAS, the increased risk for schizophrenia is 5 to 15% more than the population. Given an allele frequency of 1% for schizophrenia, a disease frequency of 1%, this means that instead of having a risk of 1% of getting the disease, you're going to have a risk of 1.2%. So it's really not a very big effect. And this graph from a recent paper in Nature shows all the variants we know for schizophrenia. And I want you to see that the vast majority, these are from the GWAS here, are down here with very, very small effect sizes, while only a few are up here. In most of those, there's some very rare mutations. This is the frequency of the x-axis, so there's some very rare mutations that cause the disease in a number of different genes. And in yellow, there is all these repeats popping under these big insertions, deletions that are mediated by repeats, and they're often de novo. And that's why they're a little bit to the right. They have a little higher frequency than the mutations because they happen de novo very often. Balancing selection in the end, and I have a question mark because this is the only study and it's been a little debatable about its validity, but what this group did, this is a group from Iceland, from the Code of Genetics, is that they took a large number of people from the population, and they had people that have they have two groups: the people that were members of creative societies, and there was a definition of what that would be, that would be artists, painters, writers, and so on, and people that were not. And they compared polygenic risk scores. We're going to talk in more detail about what these are, but in a nutshell, it's about the number of risk alleles you carry for the disease from the GWAS, the variants that we know about. So the more you carry, the higher the score is, the less you carry, the lower it is. And they found that people that belong in creative societies actually have more schizophrenia risk alleles than people that don't. So they say there is still an advantage to have the alleles of schizophrenia as long as those as you don't get when it becomes a disease. So all these mechanisms should be at play, and it's very likely that they are. So I want to jump here to go from genetic variation, how it's important to phenotypes, because genetic variation, if it has some functionality, then it will lead to different phenotypes, and they're not necessarily disease phenotypes. It could be any phenotype, like height, hair color, skin color, blood pressure, and so on. Or it can be abnormal, like obesity, diabetes, hypertension, and I'm talking about common disorders here because I'm talking about common variation. Now, phenotypic variation can be either continuous, like most of these traits, like height, but hair color is somewhere in the middle, skin color is more or less continuous, blood pressure is continuous, or it can be binary, like, and again, that's depends on the definition too, but the high blood pressure is a binary phenotype, although it's based on some threshold. So the same for obesity and so on. Now, in quantitative, so we're going to talk a little bit about quantitative traits, and I wanted to get you through some basic statistical concepts so that make sure you understand them. One is the sample distribution, and this means, so imagine on the x value, on the x-axis, the value of the trait, of height, different height, centimeters of feet, whatever you prefer, on the x-axis. On the y-axis is how many people, and these are bins like the square parts of bins, so like from this height to that height, you have that many. And as you make the bin smaller, you can end up with a smooth curve. So this is the distribution of the trait. How many people have this value, how many people have the other value. And when the distribution looks like this, we will see that's a normal distribution, but we'll get to that later. Then there's the sample mean, which is usually noted by an X with the line, horizontal line on top of it. That's the mean of the sample, which is simply the sum of all the heights of this 100 people, for example, that we measure, divided by their number, divided by 100. So this more or less tells you like what the average, as we say, is of that trait. An important knowledge is also the median, which is similar to the mean, but instead of adding up the values and dividing them, you try to find the place in the curve where you have half the observations on one side and half on the other. So you say that the median is 5 feet 7 for this population because 50 of them are more than 5 feet 7 and 50 of them are less than 5 feet 7. That's the median. In a normal distribution, usually the median and the mean will be in the same place. But if this was not symmetrical, if it was skewed, then the median would go toward the skewed side. So that's why they're more important in understanding the quantitative trait. Another thing to know is what the variance is. And the variance is a measure of really how much does the trait vary, how variable is the height. If you look at dogs, how variable their size is, it's like, compared to humans, it's hugely variable. So you need a way to compare that and see how you can measure it. And so variance is measured by what is symbolized by S squared. The reason it's squared is because what you do is you take, for each person who has a height x, you subtract this person's height by the average height, the mean height. So you see how different is this person from the mean. And then, because you care about the variance, but not about the direction in which the person varies. So if the mean is 5 feet and one person is 5'2" and the other person is 4'8", they're both equally distant from the mean. So you measure them both as equally variant. You don't care about the direction, so you square the whole thing. You square what's in parentheses, the difference between the two values, to get rid of the sign, the negative sign, and you sum all this up, and then you divide it by the population number. The -1 is a long story that you need to go to a lot of math, statistical math, to know why it's better to say than to do -1. So this is the variance, and then from the variance we also calculate what's called the standard deviation, which is practically taking the variance and bringing it back from being a square, so getting the square root of the variance. So everything was squared up here. When you do the standard deviation, you put this same formula here under a square root. That brings it down to a number that has a very important property, and that's why we like the standard deviation as a measure. And that is that if the distribution is normal, if it looks like this, then, and this is the mean, if you go 2 standard deviations away from the mean, then you know that 95% of the people are here, and only 5% are here. So it's actually not 2, it's 1.96, I think, but for all the persons, 2 standard deviations. So that makes it very useful in understanding what that variance is. That you know, so if I tell you that the average height is 5.5 feet and the standard deviation is 2.5, I'm sorry, not 2.5, is 0.25, then you know that between 4 and 6 feet, there is 95%, and only 5% of people are beyond that. Okay. A lot of lab values when you do lab tests are based simply on that. So they say it's abnormal when you're above the 95% of the population. And so it might not really mean anything, but it's a flag that you're not near where everybody else is, so it's something to look at. But I want to talk about the variance because I want to talk about the components of the variance, which are going to help us understand variability next. So the variance of a quantitative trait can be split up into 3 different things. The genetic variance, which I have as underscored with a mark with a G here. The environmental variance, so this is the variance that is due to genetic factors. The variance due to the environment, and the variance due to the interaction between the genetics and the environment. Okay, so this is a total phenotypic variation, and then the definition as I gave them up there. So the genetic variation itself can be further subdivided. So here we're talking about just genetic variation, but it can be the additive genetic variation. So these are variants that the more you have of the risk allele, the more they add to the risk added. So if this one adds 20% of the risk, the other adds another 20%, and the other adds another 20%, overall we have 60% of the risk. There's the dominance genetic variance, where it's similar to the additive, except that if you have one allele, it adds the same to the risk as if you have 2 alleles for a given variant. So there's a dominant effect, which means that the second allele doesn't add anything more. So it needs to be calculated a little differently. And there's also the interaction genetic variance, where you can have epistatic effects, where an allele is only risk allele in the presence of another risk allele, and it's not risk allele in the absence of another risk allele. So these two, the dominance and the interaction variance, are very hard to calculate. That's why people most often stick with the additive variance. This doesn't mean that they don't point that out. So this brings us to heritability, which we see that there is the capital A heritability, or the broad sense heritability, and the lowercase a heritability, or the narrow sense heritability. And the difference between those two is practically that the broad sense heritability calculates what the fraction of the phenotypic variance is due to genetic variance, all of the genetic variance, while the narrow sense, again, looks at the fraction of the overall phenotypic variance that is due to only the additive genetic variance. And again, the reason we study this is mostly because we can measure it much better, we can find it much better. It's very hard to find interaction in the genome. Now, I want to point out that that heritability is specific not only to the features of the population, but also to the environment in which the population is in. Because an allele might only have an effect in a given environment and might do nothing in another environment, in which case it will not participate in the heritability. And as I said, it indicates the proportion of the variance that results from genetic variation, not how genetic the trait is. That some people say this is not a very scientific talk. I just hear it all the time. So as I said, the variability of the trait in the same population but in different environment can be different because of the interaction. Inbreeding, I'm not going to go much into that. I'm just going to talk about the definition of the two coefficients of inbreeding, the coefficient of inbreeding and the co-ancestral coefficient, with the two ways we measure it. They're very similar. the coefficient of inbreeding is the probability that two alleles in one locus are derived from the same allele in an ancestor. So if you take from a population two alleles that are the same allele across, how far back do we go before they, what's the probability that they come from the same allele in an ancestor? The co-ancestry coefficient is really the same thing, except instead of taking two alleles of random from the population, you look at the two alleles of the person. Say I'm heterozygous of the variant for alleles A and B, and what is the possibility that this allele A that I had came from the same two people when you go up my family tree? Very, very similar concept. But they can be very useful for certain kinds of disease mapping, disease mapping, recessive traits, and so on. I don't want to go much detail about that because I want to talk last about population stratification, which is the existence of a population of subpopulations with different allele frequencies. We touched on that earlier. We said there isn't migration, bottlenecks, absence of random mating, and with smaller populations, you can have more drift, and rare alleles can be eliminated. Then this population, if there's no random mating, this can happen with a subgroup, and then you can have heterogeneity created within a population that originally was part of the same population. You can imagine that religion can do that. You have a population, some people turn to a specific religion and tend to mate only with people of the same religion. After a number of years, this will create them, this will make them different than the other population because of genetic drift, and that difference will lead to others. And as I said earlier, through random mating, it dilutes and eliminates eventually population structure. How do we identify population structure? Carry Weinberg is the first thing that we mentioned. If you're doing GWAS, what you will see if there's that mixture is a very high number, a very big increase in positive signals. I will explain later why. And that can be visualized doing what we call Q plots, which are plots where you plot the expected p-values versus the observed p-values. Ideally, you want to see that only at the very tail there's some going above the middle line where you have the real positive signals. If you see the expected always, or if you see the observed always being a little higher than the expected, this means you most likely have a problem with that mixture. And there's a number of ways to correct about this. This lecture I definitely have a number associated with it. The good news is that when you have large-scale population data, as we do in GWAS, you can do two principal components analysis because the subgroups will have strongly correlated genotypes with each other, and population PCA picks up those correlations and picks up those subgroups, so then you can actually graph those principal components and see that people separate based on their genetic ancestry. So then you know, you can see that you have an average sample. Not only that, but it gives you a measure of membership in the population, so you can correct for that and still use your data despite the stratification. Now, I'm going to finish with explaining why you get false positives by population stratification. So I want to just want to give an example. So let's say that you have an allele, you have two populations, you have a disease that is more frequent in population B than it is in population A, with a 4% frequency in population A and 16% frequency in population B. But you don't know these two populations, so you go and recruit people for your GWAS from these people. Now, let's also say that for a specific variant, this is probably going to happen for many of the variants in the genome, the two populations also have allele frequency differences, where population A has allele frequency for that specific allele of 0.3 or 30%, and population B has a frequency of 0.5 or 15%. So what happens if you go and recruit these people without any knowledge of the two populations? Let's say you want to recruit 1,000 cases and 1,000 controls. So 1,000 cases will contain 200 from population A and 800 from population B. And that's because the disease is 4 times more frequent in population B. So by recruiting by disease, you're recruiting population B. The reverse happens when you recruit your controls. By choosing your controls, you will choose slightly more from population A than from population B, because population A tends to have less disease, therefore more healthy controls. And this, so it will contain, instead of 500 people, it will take 534 about from population A. You can do the math on that if you want. So the allele frequency in controls will be 0.39, while the allele frequency in cases will be 0.46. So this means that you will think that there's an allele frequency difference between cases and controls for that SNP, and you will wrongly assume that this SNP is associated with disease, when this SNP is really not associated with disease, it's associated with population, and the population is associated with disease. So this will happen in thousands of SNPs across the genome, and will give you all these false positives. And that's why that happens. And I believe this is my last slide. This is my last slide. So I just say, why does it matter? Because in GWAS you will get false positives. In the past, we used to do this equilibrium test. We'll talk about that in the association lecture next week. But now we just can do PCA. And now we have a few minutes of questions. And also, if you minutes will tell me if things are not clear, I would love to know. If somewhere is lost in it, or I'd like to know that.
I'll share that decode paper that you put in the slides with everyone as well.
Yes. Oh, you share it? Okay.
I'll share it.
Whenever you were talking about moving between,
Can you speak up a little?
yeah, sorry. Whenever you were talking about like moving between, for linkage complete like versus perfect linkage disequilibrium. Once you reach like the 2 haplotype, like do you just assume that you're not going to get a recombination event that can split you back to like the 3 haplotypes, or you're less likely to go like backwards?
So once you reach the 2 haplotypes, you will need the new mutation, because the other one is not there anymore to recombine. Or you will need another population where the other haplotype is still present. That's part of the drift as well. When something is lost, it's lost. When an allele is gone to 0, or the reverse when it's gone to 100%, the other allele is gone to 100%, that's lost. And unless it's still present in another population, it's gone forever. And it's why preserving diversity is important, because you might be losing alleles that can be beneficial under circumstances that we don't know yet. There was a potato famine in Ireland in the 1920s, I think, where what they had done in Ireland, they were selecting the potatoes that were growing better, that they liked most, so they could end up growing only one kind of potato on the island, and then a disease came in, and it eliminated all the crops, and there was a famine and people died because they didn't have the other. And ever since, in agriculture, people have plants with seeds to make sure they keep diversity, so that if something happens, they can go back.
Remind me what your exam schedule is next Friday?
We have a pathways exam from 9 a.m. to 11:30.
That's just way too long for an exam.
We really,
Yeah. I can't run too fast anymore, but is there any chance that we can invade your spring break for the Applegate Liquor?
I think some of us won't be here.
Is there anybody who won't be here?
I won't be present in person, but I could come on Zoom.
You can Zoom as well?
I can Zoom.
All right, so we'll try to find a time, and it may have to be that week. But of more importance, the first recipient of the Leslie Lecter Award for Outstanding Educational Administration will be announced today. And I won't tell you who it is, but Sandy's at the award luncheon as we speak.